Sandwich ELISAs are widely used in clinical and research settings for detecting antigens in crude biological samples. This video demonstrates how to perform a sandwich ELISA using Protein Tech's mouse IL-6 ELISA kit. Be sure to check your kit-specific protocol for each assay. Allow all buffers to reach room temperature before starting the assay, and make sure all precipitated salts are fully dissolved. All Protein Tech ELISA kits come with microplates pre-coated with capture antibody, ready for addition of samples. Return excess microplate strips to the foil pouch and store at 4 degrees Celsius for future use. Label the strips that you need for your experiment. Reconstitute the vial of protein standard in the corresponding sample diluent, as instructed in the product datasheet. Serially dilute the protein standard according to the manufacturer's guidelines to create known standards for a standard curve. Start by adding 500 microliters of sample diluent into labeled tubes. Next, add 500 microliters of reconstituted protein standard into the first tube and mix thoroughly. Transfer 500 microliters of this mixture into the next tube. Continue serially diluting your standards in this way, mixing thoroughly and changing pipette tips between tubes. Add 100 microliters of all standard, samples, and controls to your ELISA plate, in duplicate or triplicate. Dilute samples in the same diluent as the standards. Make sure sample addition is uninterrupted and completed within 5 to 10 minutes. Include a sample diluent-only control for your blank reading. Seal the plate with a cover seal, pressing it firmly over the microwells. Incubate the plate for 2 hours at 37 degrees Celsius. Dilute the 20 times wash buffer concentrate in deionized water, as instructed in the product datasheet. Gently remove the cover seal and discard the liquid from the wells by decanting into a sink. Wash by adding at least 400 microliters of wash buffer per well. Repeat the wash step 4 times with fresh wash buffer. Following the last wash, firmly tap the plates on fresh paper towels to remove residual wash buffer. Avoid letting the wells dry out completely. Dilute biotinylated detection antibody in the detection diluent according to the product datasheet. Add 100 microliters of detection antibody solution to each well. Seal the plate with a cover seal and incubate for 1 hour at 37 degrees Celsius. Please note, some protocols will require an additional incubation step with a secondary antibody. Repeat the wash step as previously described. Dilute the Streptavidin-HRP in detection diluent according to the product datasheet. Add 100 microliters of one-time Streptavidin-HRP solution to each well. Streptavidin binds with very high affinity to biotin. Seal the plate with a cover seal and incubate the plate for 40 minutes at 37 degrees Celsius. Repeat the wash step as previously described. Add 100 microliters of the TMB substrate solution to each well, keeping the solution protected from light. The substrate solution should remain colorless until added to the plate. Cover the plate with a cover seal and incubate for 15 to 20 minutes in the dark. TMB acts as the HRP substrate, and during the incubation, the solution color will change from colorless to blue. To stop the reaction, add 100 microliters of stop solution to each well in the same order as addition of the TMB substrate. Addition of the stop solution will change the color from blue to yellow. Mix by tapping the side of the plate gently. Avoid skin and eye contact with the stop solution. Immediately after adding stop solution, read the absorbance on a microplate reader at a wavelength of 450 nanometers. If possible, record a second wavelength readout at 630 nanometers to perform wavelength correction. Protein Tech's online ELISA calculator can help with your data analysis. This tool will calculate the average of the OD values for each standard and sample replicates, and will subtract the average of the blank readings. Select the appropriate regression model and a standard curve will be generated, with the concentration on the x-axis against the absorbance on the y-axis. The concentration calculator will then interpolate the unknown concentrations from the standard curve. Remember, if the samples have been diluted, the final concentrations must be multiplied by the dilution factor used.